Hello everyone, introduce myself. My name is Ahmad Rifi Alfian from King Mongkut University of Technology Tongguri. Here I'm going to talk about my presentation by title Production of Sophorolipid Biosurfactant by Candida riodocensis yeast using hydrophobic substrate. First of all, I would like to divide my presentation into introduction, materials and methods, results and discussion, and conclusion. Biosurfactants are widely known surface active agents of microbial origin produced by bacteria, yeast, or fungi. They are amphiphilic compounds with a hydrophobic and a hydrophilic moiety that tend to interact with phase boundaries in heterogeneous systems to solubilize the organic substrates. These compounds comprise a wide range of chemical structures such as fatty acid, lipoprotein, neutral lipid, phospholipid, and glycolipids. Phospholipids are widely known studied and explored from glycolipid class. And for these compounds are common produced by Candida, mainly Candida species. For our study, we focus on Candida riodocensis as a model to produce sophorolipids. And then what are sophorolipids? Sophorolipids are secondary metabolites secreted in the stationary phase under nitrogen limiting conditions. For sophorolipids, as we can see here, they are divided into two forms, sophorolipids in lactonic form and sophorolipids in acidic form. For lactonic form, they are divided into monomeric lactone and dimeric lactone. Nowadays, many applications of sophorolipids we can find in the market are several applications such as detergent industries, pharmaceutical, microbial enhanced oil recovery, and cosmetics. They have attracted significant attention of natural surfactants because of their numerous advantages over chemical and synthetic sophorolipids. These include low toxicity, high surface activity, good environmental compatibility, and biodegradability. How does Starmella mobicola produce sophorolipids? Sure, for the production of sophorolipids, they depends on some genes that contribute to sopholipid production. Here we can see like acetyltransferase, lactone esterase, glycosyltransferase 1 and 2, and also cytochrome P450. Yeast can produce sophorolipids depends on carbon source and nitrogen source. Both of them participate in cell growth in case of metabolism, and another one is building blocks for biosurfactant biosynthesis in case of production. For hydrophobic substrate have, have gained attention because they have an ability to promote sopholipid synthesis. For the objective of this study is to characterize the properties and potential of sopholipids using palm oil as a hydrophobic substrate and glucose as a hydrophilic substrate. Here is the materials and methods for this strain. Candida riodocensis was procured from the collection of Gene Technology Laboratory, ChemUTT. and then we stick in the agar plate and incubate for 24 hours at 30 degrees Celsius, and then we remove to seed culture. For the production of sophorolipids, we use 50 ml of media contains hydrophobic substrates or palm oil, glucose, yeast extract, and urea. After we produce sophorolipids in the media during fermentation time, and then we estimate the yeast cell weight and sophorilipid production. And for sophorilipid estimation, we use separatory funnel is to separate the fermentation media with ethyl acetate. And then for rotary evaporator, we use to remove ethyl acetate and hexane and also some of metabolites produced during fermentation. For the last step of sophorilipid estimation, we weigh the yield of sophorilipid by gravimetric methods. And then for dry cell weight, we wash by the water twice, and then we filter. After that, we dried the yeast cell into incubator at 80 degrees Celsius overnight. For the purification of sophorolipids, we elute our sample into silica gel column that contains chloroform methanol using a gradient system. After that, we analyze the fraction with chloroform methanol as the mobile solvent. For the characterization of the purified sophorolipids, we use FTIR. So for this analysis, we will get the group of compounds that they are from our samples. Okay, this is our results and discussion. The screening for biosurfactant producing yeast, we can use oil displacement activity as preliminary experiment. So for this experiment, we can predict whether our strain are able to produce sulfurolipid or not. So for this experiment, we will measure the activity of oil displacement by Candida riodocensis. So for the media, we split it into two parts or two forms. First one is negative treatment and the second one is positive treatment. So for positive treatment, we add oil as a hydrocarbon source. So for negative treatment, the media is without oil. So for this figure, we can see that positive treatment has ability over negative treatment, which means hydrophobic substrate can promote the production of sophorolipid in media. For this figure, we investigate about the biosurfactant production by Candida riodocensis grown at 30 degrees Celsius with 150 rpm. And for the results, we can see here that the highest biosurfactant yield of 45.70 gram per liter was obtained on third day of cultivation. It was comparable with the yield of 32 gram per liter by Stanmela bobicola under similar media conditions. Here, we wonder about the biomass that come from oil content and no oil content, or we can say without oil content. So for this result, we can see that the differentiation about the biomass from oil content in the media and without oil content in the media. So the yeast cell concentration was overall higher when palm oil was supplemented to the culture media. This is the physiochemical characterization of biosurfactant. We compared our result by previous study here. Sophorolipid S is the standard from previous study, and then Sulfurolipid Candida riodocensis is our sample. For the standard, they used sulfurolipid sulforolactone. So for this result, we can compare about the lactonic form of sulfurolipid. The retention factors of 0.68 and 0.78 are comparable to retention factors of 0.49, 0.56 and 0.68 identified as lactonic sopholipids by previous study. Also here, we can see one spot with an RF of 0.18, similar to retention factors of 0.13 and 0.9 reported as acidic. This is the result of FTIR. So we get several functional groups in our samples, such as alcohol, methylene, esters, alkanes, carboxylic acids, lactones, and sugar. We consider that our compounds contain lactone and acid, which two functional groups here are commonly found in natural of sophorolipids. For the conclusion, Candida riodocensis is sought enhanced ability to synthesize biosurfactants in the presence of a hydrophobic substrate in the culture. The biosurfactants from Candida riodocensis were characterized and identified as sophorolipids containing both a lactone form and an acidic form. And then this strain exhibit robust sulfolipid production with a yield capacity that is comparable from the previous study by using Salmonella bombicola. I would like to acknowledge to Petra Prajomklau Master Scholarship funded by King Mongkut University of Technology Thonburi, and then Thailand Science Research and Innovative. We also thank to our division of biochemical technology and then our lab to support our experiment. And then I would like to say thank you for TSB conference that give me to attend this conference. Thank you very much.